Blood is a witness to our health. It speaks when we learn to listen. Welcome to AHS Guide, your trusted companion for mastering the 5 core subjects of allied health sciences as per Rajiv Gandhi University. Here, we make complex concepts simple, clear, and exam ready, helping you learn smarter, remember longer, and build a strong medical foundation. In today's lecture, we'll explore one of the most essential laboratory parameters in hematology, which is hemoglobin estimation. The various methods of hemoglobin estimation, which include Sahli's method, Cyanmethemoglobin method, and other commonly used techniques. The normal hemoglobin values for men and women and children, and how the deviation from these values indicates conditions like anemia and polycythemia. By the end of this session, you'll have a clear understanding of how hemoglobin is measured in the lab and the clinical significance of normal and abnormal values. The first topic for today, which is hemoglobin estimation, different methods and normal values. The hemoglobin estimation is a process of determining the amount of hemoglobin present in the blood, an important diagnostic parameter for anemia and polycythemia. Methods of hemoglobin estimation, which includes Sahli's method or the acid hematin method, Cyanmethemoglobin or Drapkin's method, Oxyhemoglobin method or automated methods are the different types of methods used for estimating the hemoglobin in a red blood cell. So these hemoglobin estimation are used as a parameter to diagnose certain diseases, blood-related diseases like anemia and polycythemia. Now, Sahli's method or acid hematin method works under the principle of hemoglobin in blood is converted to acid hematin by adding a normal 10 hydrochloric acid solution. Now, the brown color of the acid hematin is compared against the standard color scale in a Sahli's hematometry meter to determine the HB concentration in the sample that you have collected. So the requirements to conduct a Sahli's method is Sahli's hemometer set, a calibrated glass comparator tube which has gradations on it which determine the gram per deciliter calibrations, and the color comparator standard as well as a stirrer or a glass rod. We have 10 normal hydrochloride solution, hydrochloric acid, and a capillary pipette of 0.02 ml marking and distilled water. So these are the requirements for a Sahli's acid hematin method. Now the principle as such explains that it is used to convert the acid hemoglobin in the blood to acid hematin by adding the hydrochloric solution into the blood sample. It is stirred and compared along with these scales that are present to both the sides of the hemoglobinometer. So the Sahli's hematometer consists of a calibrated glass comparator tubes on both the sides which have gradations of gram per deciliter, and the color is also present along with these tubes. There is a stirrer present, the glass rod that is placed here. It is used to stir the normal 10 HCl solution along with the blood sample. Then there is a capillary tube which is used, capillary pipette which is used to draw in the amount of blood that is required to conduct a hemoglobin estimation test. Here this pipette is 0.02 ml, has 0.02 ml marking, and you can also use distilled water to clear the tube once the test is conducted. Now the procedure that has to be followed while performing a Sahli's method are fill the comparator tube by adding the normal 10 HCl solution in the graduated comparator tube up to the lower marking that is present in the tube. Later, collect the blood sample using a capillary pipette, and you are supposed to collect around 20 microliter of blood through the pipette and wipe the outside pipette to remove the excess of blood that is flowing out of the pipette. Now mix the blood and acid in the acid-filled tube, stir using the glass rod, then allow to stand for 10 minutes after the stirring is done so that the HB gets converted to acid hematin. Dilute the water, add a distilled water drop by drop while stirring until the color of the solution matches the standard color in the comparator. Once the standard color is obtained, read the value and note the level of fluid in the graduated tube against the HB scale, which is in the scale of gram per deciliter. So when you are diluting the water, you're supposed to add drop by drop and stir the solution with the glass rod. Once there is a standard color that is represented in the comparator, until and unless you get the standard color, you're supposed to keep on adding the distilled water into the mixture of blood and HCl solution, 10 normal HCl solution. Once you obtain the standard color, compare the amount of liquid inside the graduated tube. So once you see at what level the fluid is, that is the estimation of how much hemoglobin is present in the patient's blood. So the advantages in Sahli's method is it is simple and not so expensive. It requires a minimal equipment. There is no major equipment that is which is costly, and maximum equipment is required. A minimal amount of equipments are required, and it is useful in field surveys and primary health centers. So it can be used in a small scale setup for healthcare. So like in field surveys, if you're conducting a hemoglobin estimation, you can use it as a predictor of how much hemoglobin is present in the normal population. Now the limitations here are, they are subjective method, that is it depends on the color. The color that you observe could be darker or lighter based upon different eyesight or different perception. So the observer error is possible. It is not very accurate because it can happen plus or minus 10% the actual error possible. So the turbidity in the blood can also cause the limitations to the HB estimation. It might be due to improper mixing effect, or the results can be varying. The standard color may fade over time. Once it has attained certain color, it might fade due to the reactions happening inside the solution that is being mixed. So WHO does not recommend this method for clinical use, and it recommends only Cyanmethemoglobin method, which we are going to see next. Now the normal hemoglobin values that are obtained in Sahli's method for men are 13 to 18 gram per deciliter, and for female it is 11.5 to 16.5 gram per deciliter. Moving on, the second method of hemoglobin estimation, which is Cyanmethemoglobin method or Drapkin's method. The principle of this method is the HB is oxidized to methemoglobin by potassium ferricyanide. This methemoglobin then reacts with a potassium cyanide to form a Cyanmethemoglobin, a stable colored compound, so it does not change its intensity of the color, and with this intensity of color is directly proportional to the amount of HB concentration and is measured in a spectrophotometry at 540 nanometers. This is a reference method that is recommended by the World Health Organization for the HB estimation. So the first thing that the principle tells is that the hemoglobin is oxidized by the methemoglobin and the potassium ferricyanide. Once you mix potassium ferricyanide and a methemoglobin, you obtain the product Cyanmethemoglobin, which acts as a stable colored compound. So this stable colored compound is going to estimate the intensity of the color that is directly proportional to the HB concentration that is present. So you can determine the HB concentration by measuring it with a spectrophotometry at a 540 nanometer. Now the reagent that is used to determine the hemoglobin present in the human body is the Drapkin solution. So this Drapkin solution consists of potassium ferricyanide, which oxidizes the hemoglobin to form methemoglobin. This potassium cyanide converts the methemoglobin into Cyanmethemoglobin. The potassium dihydrogen phosphate is the buffer that is formed during this reaction, and there is a non-ionic detergent that lyses the red blood cells. So once the red blood cells are broken down, the hemoglobin is released into the solution. by the non-ionic detergent. Now, the potassium ferricyanide plays an important role in conversion of hemoglobin to methemoglobin and finally into the cyanomethemoglobin. This while the formation of cyanomethemoglobin, there is a buffer that is formed, buffer which acts as a carrier to the reaction. The potassium dihydrogen phosphate is the buffer. Now, the procedure of this cyanomethemoglobin method is by taking 5 ml of Drapkin solution in a clean test tube. You can add a 20 microliter of blood, which is done by micropipetting. After you micropipette blood and add a 5 ml Drapkin solution into the clean test tube, mix gently and allow it to stand for 5 to 10 minutes at room temperature for complete conversion. Once the conversion is done, you can observe, see the or measure the solution under spectrophotometry at 540 nanometers. Now, comparing this standard cyanomethemoglobin solution or read directly using the calibration that is present on the tube or the spectrophotometry. Now, the advantage of this method is that it is most accurate and reliable method, and it has the World Health Organization reference standard. This method is used to perform on all forms of HB except the sulfur hemoglobin, because it has a sulfate conjugate along with the hemoglobin and it is difficult to determine the under spectrophotometry. The advantages of this method is it provides a stable colored product and it does not depend upon the observer's error. And the disadvantage of this method is that it requires a spectrophotometry or the photoelectric calorimeter, which might not be available all times to determine the hemoglobin estimation. Now, it uses some toxic product reagents like potassium cyanide, which can be very difficult to dispose of and to handle it also. Cyanide is like a poison kind of product that is being added to determine the hemoglobin, and this method is slightly longer compared to Sahli's method because it requires the settling and reaction of the hemoglobin along with the potassium ferricyanide. Now, the normal values that can be obtained in this method for men is 13 to 18 gram per deciliter and for women 11.5 to 16.5 gram per deciliter. Now, the oxyhemoglobin method is a method that is used to estimate hemoglobin. Now, the principle of this method is that HB in blood is converted into oxyhemoglobin by exposing it to oxygen. The color intensity of oxyhemoglobin is then measured under the calorimetry or spectrophotometry at a wavelength of 540 nanometer. The color produced is directly proportional to the hemoglobin concentration. Now, the requirements of this method is diluting solution like ammonium oxalate solution, which is involved in hemolysis of red blood cells. Then you need an oxygen source in order to expose oxygen into the blood and cause the formation of oxyhemoglobin. Then you need a calorimeter or a spectrophotometer to determine the wavelength of the color intensity of the hemoglobin. So this procedure involves collection of the blood sample. It can be either venous or capillary method to obtain the blood sample. You can dilute a small amount of blood with ammonium oxalate solution to cause the hemolysis, followed by exposure of oxygen or dilution of the sample with oxygen by shaking in air or passing oxygen through the blood sample. Once the blood sample is exposed to oxygen, it forms oxyhemoglobin, and the oxyhemoglobin can be used to measure the color intensity under spectrophotometry. It has a which is above 540 nanometer intensity, and you compare the absorbance with the standard solution to determine the HB concentration. Now, the advantages are fairly simple, does not involve any toxic chemicals. The disadvantages is it is not very accurate and is affected by the turbidity and the incomplete oxygenation. The blood can form lumps, or there is not proper oxygen that is delivered into the hemoglobin, and it does not measure the abnormal hemoglobin derivatives like carboxyhemoglobin and methemoglobin, because it has a carbon dioxide attached with a hemoglobin, or a methane group that is attached to hemoglobin. But this test refers to the use of oxygen combining with hemoglobin to form oxyhemoglobin. So this can be a disadvantage to carry out the procedure or the method. Now the largely obsolete and replaced by a Cyanmethemoglobin method, because Cyanmethemoglobin is a standard procedure that is recommended by WHO, but this cannot be recommended because of various disadvantages that it holds. The normal values that are obtained by this method is, for men it is 13 to 18 gram per deciliter, and for women it is 11.5 to 16.5 gram per deciliter. So now moving to the last type of method that is used to determine the hemoglobin estimation is the automated methods, or the machines that we use in our daily blood sampling and testing. The principle of this automated hematology analyzers are measuring the hemoglobin concentration using a calorimeter or a spectrophotometer principle. These are usually based on the Cyanmethemoglobin or the sodium lauryl sulfate methods. So the modern analyzers break down the red blood cells, convert it into hemoglobin into a stable colored compound, and they measure the absorbance of specific wavelength by the hemoglobin and determine the hemoglobin present inside the blood. Now the common automated methods are Cyanmethemoglobin method, which is integrated along with the machine. So it works as same as the principle of Drapkin's method, but it is performed automatically by the analyzer. So the same method is adopted by the automated Cyanmethemoglobin method. Here it requires cyanide content reagents, which is again a toxic solution that you are adding. Now SLS is a sodium lauryl sulfate method. This is most widely used among the modern analyzers that are adopted in the laboratories. The principle of this method is that hemoglobin reacts with the sodium lauryl sulfate reagent and forms a sodium lauryl sulfate hemoglobin complex. This complex is measured under a photometry at 555 nanometer, and the advantages of this method are they are non-toxic as compared to the Cyanmethemoglobin, and it is environmental safety, accurate and fast, and hence it is most widely used among the modern analyzers that are adopted in the blood banks. Now conductivity or impedance method. Here some analyzers indirectly estimate the hemoglobin by correlating with red blood indices and hematocrit, but not commonly used as a primary HB measurement. So these are dependent upon the red blood cell indices, that is the number of red blood cells that are present based upon the clotting or bleeding time of the red blood cells, or the various other methods that are required to determine the hemoglobin, like the packed cell volume and various other methods that are used, like ESR can be used to determine the hemoglobin, but these methods are not directly used to measure the HB. The steps in the automated HB estimation is the sample aspiration. So the analyzer draws a small amount of blood. There is the aspiration of the blood by the machine itself, and the red blood cells are broken down automatically by the machine, and the chemical reaction is happening wherein red blood cells reacts with the reagent to form a colored complex. Later a photometric reading is observed by the fixed wavelength that is absorbed by the red blood cells, the hemoglobin solution, and which is usually around 540 to 555 nanometer. So this wavelength is going to display a certain HB value that can be taken as a result of the HB estimation. So the disadvantages of automated methods that they are highly accurate. The advantages of these automated methods are that they are highly accurate and precise. Rapid process. It does not require manual operation for each sample. It takes around hundreds of samples in 1 hour per hour, and the measurement of HB along with other hematology parameters can also be included, like the RBC count, WBC count, and platelets can also be shown by the automated methods. No, there is no subjective error like the color perception that is being done in Sahli's method. It requires only few microliters of blood and does not rate excessive amount of blood. The disadvantages can be it is expensive instrument. It does not, and it requires maintenance and calibration every week or monthly. Maintenance should be done, and it requires power and reagent dependency as well. Now, the normal values of these automated hemoglobin estimation is for men 13 to 18 gram per deciliter, and women it is 11.5 to 16.5 gram per deciliter. Anything above or below these ranges determine that there is a defect in the hemoglobin of the patient. So that is when you diagnose a certain kind of disease. So moving on, the next topic is packed cell volume, which will be dealt in the next video. Till now we have learned about the different estimation methods of hemoglobin, the principles, the requirements of each method, the procedure that is performed. So the hemoglobin estimation, as I told in this video, there are 4 methods of it, which is Sahli's method, the Cyanmethemoglobin method, we have Oxyhemoglobin method, and finally the automated methods. We have seen the various advantages and disadvantages of each method and the normal values that is going to determine the HB of a patient. So anything above or below these normal ranges are going to cause are going to determine or diagnose the different kind of diseases that can be observed in a human. Thank you, and keep watching AHS guide. If you have any doubt, put it in the comment, as well as there are notes provided in description and a next video link that can be obtained in the description as well. Till then, keep watching AHS guide. Thank you.